Our guest today, Dr. Erin Cawston, originally hailed from the Hawke's Bay. She began her research career at the University of Otago, where she completed her degree in medical laboratory science. She was introduced to the field of neurological research through her work in diagnostic with Dr. Robin Olds, who is now Chief Executive of the government's Health Research Council. Her curiosity in brain receptors was piqued during this time, and she began a PhD, still at Otago, as a Neurological Foundation postgraduate scholar. Dr. Cawston diverted into the pharmacological side of research and decided to extend her experience by going overseas to the Mayo Clinic in Arizona. In 2011, Dr. Cawston was awarded the Neurological Foundation Repatriation Fellowship so she could return to New Zealand with her extensive knowledge in her specialist area of research. The extremely hard work during her fellowship year in 2012 was rewarded earlier this year when she was awarded the Morris Peikel Postdoctoral Fellowship from 38 outstanding applicants, so she could build on her receptor work at the University of Auckland. Dr. Cawston is based at the Centre for Brain Research, where New Zealand's top neuroscientist, Professor Richard Fall, oversees 54 laboratories and 300 brain researchers. Taking a day out of her research work, please join me in welcoming Dr. Erin Cawston. Dr. Cawston, can we start with a few facts about the brain? Some simple specific about the physical brain.
Well, the physical brain is basically this large spongy tissue. It's about 3 pounds, so 1.5 kg. Interestingly, this one structure holds over 100 billion neurons and many other accessory cells. And within this one organ, 20% of our energy metabolism is taken up just to supply energy for this one region of the body.
Amazing. And can you just dispel a few myths about the brain? My favourite is we only use 10% of our brain.
That old one. Well, when you think of it in a purely biology stance, if we had a brain and we only use 10% of it, that means 90% of that brain is not being used. We're using all our energy metabolism for something that we're not using. So unfortunately, it makes no evolutionary sense that we're not using 90% of our brain. Recent early work has shown that with fMRI and PET scans that basically everything that we do in a normal life, just you sitting there, emotion, keeping yourself upright, all the different parts of the brain will be firing. So we use the majority of our brain the majority of time, even just for simple things like sitting, like our proprioception for where we're sitting, breathing, emotion, anxiety, all those different things basically fire different areas of the brain, so we're continuously using it. You can, however, increase your capability of using different parts of the brain by, brain activities and things like that. But, the myth of 90% of the brain not being used is purely false.
What about left-hand, right-hand dominance?
It's a similar sort of thing in the sense that for years people thought the right was more creative and the left was more analytical and logical, and you could put sort of things into a basket. But now with our technologies and things that we know, it's purely, we know that both sides of the brain are crucial for just every part of our living. Yes, some are a little bit more predominant in different areas, but you know, you can't lose one side of your brain and basically still carry on. So it's all works together in a very complex environment. So we can't just put one side being the predominant over the other.
I think one question many of us in the audience will be interested in is that thought that it's all downhill for the brain after 40.
Once again, it's one of those things that, yes, after 40 or in older age, some facets of our cognition decline. You talk to any youngster and they'll be able to remember names really well. They'll be able to identify, if you put certain objects in front of them, which came first, and if you basically never have a staring competition with a 10-year-old, I'm just telling you now, that they're always going to win. It's basically there's certain facets of a younger brain that are still developing and able to be acting more of the time, but in older age, or with maturity, one might say, you also gain many skills. So increase in vocabulary, increase in perception of the world, so you're able to empathise more with situations. You've had the life experience, so you're actually, if anything, gaining skills with age as well. You're able to interpret whether just by sort of pure first interaction with someone, if you trust them or not. Those are skills that younger people don't have. So, you know, the myth is false and true in a sense, but mostly false, because I think, you know, you can be healthy and active and the brain can still be firing on all cylinders after 40, well and truly.
Is the brain hardwired?
This is sort of from a very earlier on stance on research, and basically the brain was thought of as a circuitry and like an electrical circuit. And yes, it is true that certain parts of the brain are wired up in specific ways. And I think the biggest reason why people talk about hardwiring is the fact that when you have cells or neurons in the brain, certain chemicals are released, and they basically change the electrical input through the brain. And so that, in a sense, is an electrical impulse, so therefore people think of wiring and electricity. In a sense, the brain is highly structured in circuitry, but it is far more complex than a basic circuitry of an electric circuit board. And it's also able to be modified. So you can get, when something is damaged, there are ways in which your brain can maneuver itself and change the synaptic impulses. So it's not really that true.
If we incur brain damage, is it always going to be permanent?
Well, this is one of the things that we know so much more now, and depending on, or every case is an individual case, and depending on the type of injury will depend a lot on treatment and outcome. And in certain instances, disease will deteriorate a region of the brain. But in the case of stroke, there have been a lot of work done in rehabilitation and basically retraining of the brain to reconnect up its circuitry and enable you to use different neurons or different pathways to actually produce a similar outcome. So not all brain damage is permanent.
Well, that's, I think, really wonderful to hear. Can you tell us now a little bit about brain research? Because, of course, that's what your whole working life is about.
Well, for a start, just seeing everyone here makes it all worthwhile, I just have to say. Brain research, especially in New Zealand, is just a wonderful place to actually be. We have so many amazing researchers working on things, and the day-to-day life is hard because there's so much we still need to know. The brain is the most complex tissue in the body. It basically, it runs us. Without our brain, we don't have anything really. And so there are so many things we still have yet to discover, but there's so many things we know now that we didn't know before. So it's sort of one of those things that you're never bored. Every day is a new day, and every day you find something more and more interesting and that can help in the future.
And what is neuroscience?
Well, it's sort of coined from the term neural science, so anything about the nervous system or the neural system. So it can encompass many different parts of the actual profession. So you can have people that are interested in spinal cord areas, auditory, sight, but there's also the neuroscience where you have, years ago it used to be just basically an arm of biology, but now we have so many different fields working on it, you'll be amazed. We have engineers, we have statistical analysis, epidemiologists working in here, neuropharmacologists, just biologists, bioengineers trying to basically uncover new ways and therapies. It's a massive spectrum now, and it encompasses basically anybody who is working on the brain, basically.
Okay. Now, I think you're described as a neuroscientist. Can you just explain what that actually means?
Well, once again, it could mean anything in the study of the brain. For me, I'm more a cell neuroscientist, so I look at the different cells making up the brain, but also I'm termed a neuropharmacologist as well, because I look at the different molecules that signal within the brain and how that affects, that is affected in disease. Also how different therapies may be able to help with basically re-stimulating areas of the brain. So I'm interested in drugs that potentially can act on different parts of the brain and basically prevent disease or help in the disease process.
I understand you're now in your second decade of scientific research. What are the biggest changes you've seen?
I think one of the biggest changes is probably public awareness, just exhibited by everybody here today. A lot more people are aware of, there's so many different facets involved in our research. I think the technology has increased just dramatically, exponentially. We have so many more things now that when I first started out in medical research, we never even dreamed of. And, you know, just purely because of the all-encompassing neuroscience aspect as well. So you have engineers now working on different things. You have a lot of neuropharmacologists looking for different drugs that can act. So basically, I think just our advancements in leaps and bounds have been the biggest thing that I've noticed over the 10 years, and I would never have guessed we'd be where we are today compared to when I first started.
So these changes have had a profound effect on what you do as a neuroscientist.
Yes, definitely. The technologies, as I said, just, you know, everything that we do is influenced by what we're able to achieve in the laboratory. And now with so many people working on similar areas, we have all these technologies coming out daily. You just need to look at any of the scientific journals where everybody is working with a similar goal, and it is to help and you know, prevent disease of the brain. And so we've got the most ingenious technologies coming out of the woodwork from, you know, here within New Zealand, but also throughout the world. So we're very fortunate. They do come at a cost, but I just think we're so fortunate to be able to have access to those. And we have many of them in New Zealand. It's just the advancement in things like MRI and magnetic resonance has basically taken neuroscience in leaps and bounds.
Now, your particular area is cell research, and I think we'd all be very interested in just learning a little bit more about that. And can we start with the basics of what is a cell?
So basically the cell in our body, like we're made up of basically all different cells, depending on what we're looking at, that muscle tissue, heart tissue, everything is made up of cells. And so they're basically a biological building blocks. Within them is even more a complex system. Basically a cell has its own way of replicating, its own way of energy metabolism. It's like a little factory in itself. And so organs are made up of millions of these little cells all working in unison, all for the same thing. So what we're interested in, and a cell biologist, people say, well, one cell must be similar to another cell, but that is not the case. All different cells are specialised in different functions, and in the case of the brain, there's very specific neurons and glial cells and astrocytes, all that make up the brain. So we look at specific cells and their markers and how they affect the biology of disease or just normal everyday physiological systems.
And how do you see these cells, and how and what do you use to observe the differences between the cells? Because I mean, they must be just unbelievably minute.
Well, everybody's got this impression of the white lab coat and the microscope and a scientist looking down. And in part, it's actually quite true. A lot of the work I'm involved in is basically we take a cell and we grow it. But we also add in looking at different parts of the cell, different proteins in the cell, and we tag them with fluorescent tags, and basically we're able to, it's amazing, actually watch specific parts of the cell interact with other cells. We're able to stimulate them with neurotransmitters or different drugs and see what happens to the cell, which is amazing. And I use something called fluorescence resonance energy transfer, and that's really a measure of how close two molecules come to each other. And if they come close enough, we're able to measure that interaction, and we're able to see it down the microscope, which is even more amazing because you can't really see these cells. A lot of what we're looking at is so minute to the eye. So that in itself, the technology is advanced in leaps and bounds.
Wonderful. And what about receptors?
So my main area of expertise is in receptors. So when we have a cell, this round organelle, we also have these little things on the surface, and they can be inside as well. My major focus is on this receptor family that is the largest in the human body, and basically every cell we have in our body has a different member of this family of receptors called G protein coupled receptors. So you've got the dopamine receptors, the serotonin receptors. I'm interested in a receptor called the cannabinoid receptor purely because it's the most highly expressed receptor in the brain and is linked with many pathologies, but also just in everyday physiological use. And so a receptor is basically something that is on the outside, and it's a way of the cell knowing what is occurring around it. It's a way of the cell being able to monitor and send signals to other parts of the cell or other cells. So it's sort of like a little messaging system that we're interested in. And in disease, sometimes these little receptors are lost, which can create part of the pathology. And we're looking at ways in which we can actually signal through these receptors and enhance these receptors, in which to maybe prevent and be neuroprotective against disease.
That's wonderful. So that is why you focus on that, because that's ultimately the goal.
Yeah.
Okay. In neurodegenerative disease, we know certain types of cells die. Can you tell us a little bit more about this and what happens when these cells are damaged or die?
So, as I alluded to before, the brain is made up of different cells, neurons, glial cells, astrocytes, all with a very specific function. But even more complex than that, you know, with 100 billion neurons in the brain, they all have synaptic, what is called synapses, and basically they all have these little projections. So one neuron can basically touch or send signals to over another million cells. It's got so many projections. But in each part of the brain, there are also specific cells that send specific signals, neurotransmitters, and so they're important for the function of memory, movement. So there's very specified regions. So depending on the cell region that's affected by disease, is dependent on what symptoms occur and what's the overall outcome for the patient. So in something like Huntington's disease, where the cells of the basal ganglia are most affected, the symptoms are associated with deterioration of that region and then other regions. So that is why neurological disease is complex in itself, because every case is slightly different as well.
Some years ago, not that long ago, Professor Richard Fall and his team at Auckland University discovered that the human brain has the capacity to make new brain cells, which was something that they had thought just was not possible. Can you just tell us a little bit about that? Because I think that's a really fascinating discovery and area of work.
Well, for a start, there's Richard up there, who's just up there. Amazing scientist, amazing man, and amazing leader. And his research with Maurice Curtis was groundbreaking in the fact that it's long been thought that if a neuron in your brain dies, that you never get it back. But Richard's research basically showed that in the case of something like Huntington's disease, that in an area very close to the area where the cells are dying off, there is a region called the SCV, and it's basically this place where when we're born, when we're growing and developing, this is where all the neurons are basically migrating out and finding their position. So, you know, in utero, when we're in our mother's stomachs, and, you know, in development, this is the area where neurogenesis occurs, where cells are created. And basically, as we get older, it was thought that this area declines in its neurogenesis. But what Richard and Maurice found was basically that in the Huntington's disease, where you have this large amount of cell death in the basal ganglia, just very close in the SCV, there is an increase in neurogenesis. So in an older patient, there's actually an increase in new cells being developed. That is huge potential because in the case of Huntington's disease, it's so close to the area affected. So what they showed is basically that the cell death that's occurring from the disease actually stimulates the body in which to create these new cells. So the implications for further on is if scientists can tame and basically get those cells to regenerate areas of disease. So that is what a lot of scientists are working on now, how to get those new cells and get them populating an area that has been affected. So I think it has massive implications for the area of neurological science, and I know that they're working very hard in which to try and gain that ability.
And I think that was research of global importance, wasn't it? I mean,
worldwide.
It was really groundbreaking,
Yep.
wasn't it? That's fantastic. What have been the biggest obstacles to your research?
I think the biggest obstacles have been, and one might say, is the pure financial. And basically, I say that because when I talk about the increase in technology, that comes with a cost, and it's expensive. And just even for me, just getting funding in which I can carry on my research has been a hard thing. So when I've got help from places like the Neurological Foundation and wonderful people like you, I think it means so much more, because basically I know I'm getting support from individuals to help me carry on my research, and I think it makes it really personal and therefore makes me want to work even harder than I would have worked if it was from another area.
New Zealanders seem to do very well in neurological research, and I think it was Professor Fall who once told us that New Zealanders, somewhere in the world somebody's got a league table of neurological research, and that New Zealand came 20th on that list, which I thought was astonishing given the size of the population. Why do you think New Zealanders excel at neurological research?
I do think, being a small country, I think we are more passionate about our people. We want to see progression and looking at the causes of disease, but I think it brings it home for the next generations as well, that we want to do better for the future. Kiwis are not shy from a challenge, and all the scientists I work with are extremely passionate, and they're really just out there doing their best, and they're not scared. The thing is, with brain research, there is so much to know. The brain is the most complex organ in the universe, basically, and we're basically trying to understand it and then build on it and try and gain ways in which we can help people with disease or prevent disease, which is our ultimate aim. And so that is a massive challenge, but Kiwis aren't shy, and they will go for it. So I think that speaks volumes. But also the support we get from everyone just makes everything so much more real, I think, as well. And also we've got initiative, I think, that other countries, we've got the real number 8 wire Kiwi initiative and go-getting sort of attitude.
Mm. And you've spoken about the obstacles to your research, but do you think generally there is an issue of resource for neuroscience in New Zealand?
Yes, I do, but in saying that, I realise everything is hard at the moment, the economy, but I do think to keep up with the way research is progressing and the amount we're learning, if we have the technology, you know, we can go even further, and that does require significant investment.
Now, I did hear somewhere, and it may even have been from you last year, that only 7% of medical research in New Zealand gets funded.
Depending on what sort of funding agency you're going for. I think at the moment some of the running rates for our government funding is around 7% success rate for Marsden and HRC. And saying that, there's a lot of amazing scientists in New Zealand, and everybody's up on the same field, and so it just makes that work even more important.
I mean, I certainly was shocked when I heard that figure. I think I would have expected it to be much, much, much higher. What do you now know that you didn't know 10 years ago? Just a big question for any of us, isn't it? Have to remember 10 years for a start. Sorry, that's probably a little unkind.
Oh, a lot. Huge amount. I think, you know, some of the biggest things, the research that Richard and Morris have done, I think there's been leaps and bounds, and just in the areas of neurological research. I think for a long time, you know, 10 years ago, people researching into the brain, you know, everybody sort of agreed it was a hard thing to do, but I think I know now that there are so many passionate people out there trying to really get to the bottom of things. I know a lot of different things just purely because our technology has advanced so much. When I started off, it was old school technology, very, you know, bucket chemistry they would call it, but now we have the most amazing science where you can see one molecule interact with another molecule. So I think the progression of a scientist is good to see. I think in 10 years' time, you know, things will change dramatically as well.
And talking about in 10 years' time, looking ahead, what would you hope to see in neurological research? If you had a wish list.
Oh, go on, cure everything. I would hope to see more targeted therapies for disease. I would hope to see therapies in which, as I said, regeneration of areas of the brain that have been affected. You know, I would hope to see a cause for these diseases be identified and basically stopping disease before it starts. You know, that's the ultimate game, that no one suffers through something like this, that we actually find we pinpoint where these diseases are. We know so much more about these diseases now, Alzheimer's disease, Parkinson's disease, but actually being able to stop it before it begins, it would be a massive thing. But more targeted therapies, medications in which to alleviate patients' suffering, but also just basically stopping it before it starts would be on my top of my wish list.
To end this interview aspect of this morning, I'd just like to end on a slightly lighter note. What about this men are from Mars, women are from Venus? What's your thoughts on that?
Could explain a lot, but when you think of it in a purely biology stance, and when we know what we know now from different technologies such as MRI and things, the male and female brain are identical. There may be a few subtleties that are different within the two brains, but they're pretty much identical. I think a lot of it's social and just the way we've been brought up. You know, there's myths, you know, women talk more than men. Well, that's false. But people still disagree with me. You know, there's myths such as that, that women are more empathetic than men and things like that. But basically, we've all got the same abilities. There's nothing unique. We're all from the same planet. It's just how we use our circuitry. It makes us unique.
Thank you. Now we have a few minutes to take questions. Can I just remind you that Dr. Cawston is a neuroscientist, not a neurologist. So if you could confine your questions to her research or neurological research, that would be wonderful. Thank you.
Could you just talk a little bit more about dementia, please?
Dementia. Well, from what I know, and once again, I'm not a neurologist, you know, just basically the decrease in certain brain cells in the region of the brain, different regions of the brain. Dementia is also defined on other things such as, you know, dementia is a massive categorisation, and Alzheimer's disease comes under that and different areas. But at the end of the day, it's death of specific regions of the brain, and they deteriorate over time. And if you look at, you know, autopsy photos, the brain is smaller and specific regions have been targeted.
You're researching non-pharmacological healing methods?
Yes, at the moment my work is really trying to look into the cannabinoid receptors, and what we've found in the in vitro work and in our studies is that signalling through this receptor does have a neuroprotective effect. So it is our hope that if we can find some chemicals that would be able to go via this receptor, we could have more of a neuroprotective effect and basically stop the deterioration of specific cells. That would be our overall aim. So therefore not have the cell death in the first place, which would be your aim overall.
I'll start again. To what degree is brain research across the world coordinated so that there isn't too much doubling up of the same research?
That's a very good question. It is highly coordinated, especially I think in the times with Skype and things like that. But you've got to remember that I think the good thing with scientists as well is a little bit of overlap is good, because you have more minds on one area. And so you have people who are, it gives you a bit of impetus as well to be the first person to sort of find specific, you know, it'd be lovely to be the first person to find a cure or some sort of drug. So I think a bit of competition is a healthy thing in that field. I think scientists in general worldwide, especially with our technology these days, interact together. We have all our conferences together, so everybody is actually a team. They're working for the same goal, and everybody wants to be part of helping alleviate suffering or actually preventing disease in the first place. So there is some overlap, yes, but I think it's only beneficial overlap in my mind.
Do you believe that when you dream at night, the dreams that you dream come true?
I wish. I don't know a lot about the dream side of things. I do know that neural circuitry is extremely complex. Don't know if I believe it comes true, but I do believe that there is a reason why we do dream, and so I would like to know more, of course, about that area, but I'm sorry I can't give you a better answer than that.
Good morning. You mentioned being able to see individual molecules. Reminds me of molecules of emotion, Dr. Candice Pert, Bruce Lipton's work on cells, epigenetics, and then you have the going down through the cells, the quantum mechanics now of cells. All these tend to make life, apart from more interesting, the research, more difficult. Do you find that as much advancement you try and make, there are these new fields to be taken into account which add to the complexity of your work?
Definitely. But it adds to the complexity, but it also creates with all the complexity that's involved, yes, especially when you get to the quantum mechanics, when you think of a cell in general, you have nuclei, you have a whole different sort of area of issue. But what I think it does help is adds a few more pieces to the puzzle, but when that puzzle comes together, it comes together at a lot clearer level. So if you can, it gives more reason to what we're doing. I think the complexity of the brain, even just going down from the mass organ to all its circuitry and then going down to the cellular structure. And then when you go into the genetic structure and the familial side of things, and it's amazingly complex, but I do believe that helps the story, because when you find bits that fit together, it makes that research so much more important.
Dr. Cawston, if a bright young person with all the necessary qualifications wanted to make a career in neurological research, is it necessary for her to take a medical degree first, or can she get into it otherwise?
Oh, she'd definitely get into it otherwise.
And how would she go about that?
I think there are many great, throughout the universities in New Zealand, just doing basic neuroscience papers. Auckland University has a great neuroscience degree in neuroscience. Also down in Otago, my background wasn't neuroscience at all. Mine was medical laboratory science and pathology, but it gave me an understanding of the genetics, it gave me an understanding of the actual overall pathology of the body, and it allowed me to focus and hone my research into a very specific region. So I think you could do many things and then do a postgraduate in the neuroscience, but I think the criteria is probably do a neuroscience degree at something like Dunedin or Auckland, and with a lot of health science papers intertwined, and then have a real passion for it, and you can go anywhere you want.
Thank you very much. It's very encouraging.
Yeah. Thanks very much for all the work you do in this area. It sounds great. I just had a question. You talked about protective factors and the chemicals and the pharmacology that you use. Is there anything that we can do? to protect ourselves against that disease?
It's a very good question. Depending on what disease you're talking about. I was talking to my parents about this actually last night, where there was something on 60 Minutes. And I think keeping your brain active and you know, Sudoku, crosswords, there are many things now on the net or just in general at libraries, you know, reading, just keeping those facets of your life. And exercise has been shown to be extremely beneficial. Music, stimulation of the brain in all sorts of different ways, just to, you know, just enjoying things like that. Dietary, there's many links between different diets being beneficial, but I think, you know, everything in moderation, your superfoods are, and your blueberries and all those sorts of things, there's links, tenuous links to all those sorts of things. But from my mind and from what I've seen in the literature, it's an active lifestyle and basically stimulating the brain constantly, and you know, looking through old photos and reading old things, and just keeping the brain going, not having it sedentary, is a great thing to help.
Does stem cell research become part of your process as well? I know that we're able to look at growing parts of the body now. Are we able to grow parts of the brain down the stem cell track?
That's a very interesting question. I think a lot of work is going, well, a huge amount of work is going into stem cells now. Mostly, or you know, from very different angles. Some from basically putting in cells to basically recreate the tissue that is dead, but also a lot of people now, and people I know working right next to me, are trying to harness the capabilities of the cells, such as I talked about Richard's work, that are very close by, and actually get them to go to a specific region. So stem cell work is very important in this region. I think, you know, we have to come from all different angles. We have to come from the actual understanding more about the diseases and actually preventing the diseases. We have to come from the angle of therapies for those in earlier stages, and then treatment for those in later stages. So stem cells can come into many different diseases, such as MS and many different sides of things. And a lot of people are working on that, and I think it's making leaps and bounds as far as I'm aware.
Yes, my husband had a brain hemorrhage 5 years ago.
Mm-hmm.
He's got short-term memory loss and confusion.
Mm-hmm.
Will that ever come right?
Unfortunately, I can't answer that question. A neurologist is the best person to answer that question, and I know people can get you onto the Neurological Foundation. Dr. John Simcock can answer questions probably in that area. I don't know is the answer, I'm sorry.
If you'd like to see Jane Sharman just standing in there with the T-shirt, the research T-shirt, she'll tell you how to get in touch with John.
Oh, hi, good morning. I'm just wondering, my ex-husband had a stroke a year and a half ago, really active, ate raw food, you know, he was over the top. Had a massive stroke, he's 61, but we work with him intensively, my daughter and I, and we're basically told he's plateaued, but we have seen so much improvement since he was told he's plateaued and basically written off. Do you believe there's a plateauing time? And I mean, I don't, but I'm wondering if you do.
I don't unfortunately know enough about the area, but I think what you're doing for him is amazing, and I do think actually getting in touch with Jane and talking to Dr. John Simcock would be a good thing. Yeah, once again, I can't really comment on that, I'm sorry.
Okay. Thank you, anyhow.
And also, have you been in touch with the local local stroke support group? Because that's a question they're probably well able to answer.
Dr. Cawston, could I ask if you personally or other of your research colleagues, if your research encompasses such specific toxicological factors as exposure to environmental things such as heavy metals, chemicals, solvents, infections, also innate or acquired nutrient deficiencies?
Not personally, and not the people I'm surrounded by just in the vicinity of our work area, but I'm more than 100% sure that there'll be people in Auckland at the CBR working on that field of research, and you could possibly just the Auckland, did the wrong thing there. The Auckland website, the university website, I could talk to you after and give it to you, and there should be someone definitely looking at those fields, especially from an epidemiology side of things, but definitely they're very, very important fields.
Hi, Erin. I'm really grateful that you're speaking about this. I'm a believer in Jesus, and I know that in the Bible it says that he heals, although he does use wonderful people like yourself to do the research and do the parts, you know, in our bodies that need to be connected. And what my question is when we're working with patients with Alzheimer's or dementia and they've had that deterioration of the cells, is there anything we can do to improve like where they are at and help them towards getting better?
It's a very interesting question, and you know, I'd like to give you specifics, but unfortunately I'm not. I think basically, as I said, trying to just stimulate the brain. There was on TV last night, there was a good thing on 60 Minutes talking about music therapy and how music therapy, I think a lot of the therapeutic approaches people are working on now, how to actually try and stimulate and help different people. But I think it's, unfortunately, I don't have an answer for that, but I think, yeah, sorry.
I think we had two people who had their hands up, and I think they'll have to be the last two, but Dr. Cawston will be around after we finish, and you're very welcome to come in and talk to her informally afterwards. So if we could just have those last two. That gentleman.
Good morning, Dr. Cawston.
Good morning.
On behalf of us all, thank you for coming, and thank you, the Neurological Society, for making it available for you to come and share with us this information that we're also very, very focused on. We've all had some experience, I'm sure, and you've been very clear in what you've said, very specific and very honest, and that's wonderful. Moving on, my question is, the brain, we understand very limitedly, but we do understand that nerve is the passage by which the brain reaches out and communicates with our bodies and our bodies back to the brain. That is an area that seems to be a great deal of difficulty for many people, myself and many people, I'm sure. So the question is, what likelihood of advance in that area is there in the future? We're learning about the things you're doing and other things, but I don't have any familiarity with discussion of the nerve paths being developed.
Oh no, I definitely think that is, as I said, neuroscience is neural science per se, and I think a lot of people working on especially pain and nerve innovation. As far as I'm aware, I've heard many people in Auckland talk on different things such as helping with pain for nerves, because they're all connected up to the brain, and you're correct, and I think there's incredible advancement in that region as far as I'm aware. Once again, everything sort of takes time, but I think it is definitely progressing, and it is definitely a focus of many people. As I said, in neuroscience, like I've got my specific area of expertise, but there are a lot of people working on stroke and rehabilitation. There are so many facets, like as Sally said at the CBR, we've got 56 different labs, and they go all the way through from evolutionary to care of patients, through pain, through you name it, they're doing it. And so I think there's great advancement, and you know, if you think this is what we're doing in New Zealand, you imagine the bigger countries, you know, and we all work together. So I think we're going to be making some great progress in the years to come.
Okay. Hi there. I'm just wondering if you've looked into the effect of behaviour training on the brain and the changes that it can make, things like classical and operant conditioning and you know, routine training and things like that.
I personally haven't, but I do think there are many people looking into those sort of, I know of many people looking into those sort of facets, and it is quite an important part of the brain health, especially even in relation to problems with the brain or disease. Those areas, cognitive rehabilitation are a very big part. You know, we'd all like that wonder drug to cure everything, but these diseases are multifaceted, and our approach needs to be multifaceted. So we need to have different training and behavioural training for the brain, and etc. But I think the one thing I want to make clear is the brain is what we call plastic. We have synaptic plasticity, and that's the one big thing, the advancement that Richard has made, and all the scientists are realising that now. You know, we all thought that neurons went to one place and they had one function, but we know now that when a neuron is, if certain neurons have died, other neurons may come in and take their place and actually try and regain some of the original function of that tissue, and that's called synaptic plasticity, and that is the way of the future, especially for a lot of these diseases, because we know the brain is amazing, and I think we didn't realise how amazing it's actually trying to fix itself in so many ways. So we need to understand that more, but that's a big focus on the future of research of synaptic plasticity and how we can train the brain into helping cure these diseases. But if you want to know more, synaptic plasticity are the key words there, because they have massive implications for the future.
Well, that's a wonderful way, I think, to end this morning. And I'd like to give a huge thank to Dr. Erin Cawston for giving up her time today and giving us such a fascinating insight into the brain and some of her research and some of the research that's happening here in New Zealand. Thank you, Dr. Cawston. I'd also again like to thank the Dowdall Trust for their huge support of this event, Mitre 10, our wonderful volunteers without whom we couldn't hold these events, and to our wonderfully committed supporters. If you haven't already registered, would you please do so? And if you register, you can find out about future events. And also, please consider joining the Neurological Foundation and helping us in our work to fund researchers such as Dr. Erin Cawston and Professor Richard Fall. Thank you so much for coming today.